Hi, my name is Linda, and my review paper focuses on how genomic regions overcome barriers to transgenerational epigenetic inheritance. So, as we're all familiar with, genetics is the study of the inheritance and of genes and DNA, but what you might not know is epigenetics, which focuses on modifications attached to DNA that cause changes in the expression of the gene without actually changing the sequence itself. These are marks like methylation marks, histone marks, and non-coding RNAs, and can be environmentally induced from our lives. If they become heritable across three or more generations in a phenomenon known as transgenerational epigenetic inheritance, or TEI, it can completely change our current view of disease and evolution, and it will mean that even your great grandparents' environment and lifestyle factors can have an impact on your health. Theoretically, TEI should be impossible because of three main biological barriers, which is probably a good thing because we don't want our ancestors' experiences to impact us. But in reality, there's increasing evidence that TEI is happening. Most importantly, there's currently a lack of systematic reviews addressing or compiling evidence of how genomic regions can escape these barriers to TEI, and that is what my review aims to address. After screening 285 abstracts and 109 full articles, we ultimately landed on 39 journal articles that best answer this question. Looking for common trends across these studies, these types of regions that tend to escape the barriers to TEI are regulatory sequences such as introns and promoters, imprinted genes, transposons, which are just elements that can jump around in the genome, which usually causes diseases, stress response genes, and regions associated with metabolic and neurological diseases in humans. Now, looking at the three specific barriers, we found significant evidence on the mechanism of how each can be overcome. Firstly is the Weismann barrier, which dictates that even when somatic cells do receive epigenetic changes, they cannot communicate them to the germline, and thus these marks will never get passed on. We found that there are actually ways to bypass this barrier. For example, sperm cells will pick up small RNA from the epididymal epithelial cells as they travel through the epididymis as they mature, therefore gaining their epigenetic information and bypassing the Weismann barrier. Next, there are two waves of reprogramming where epigenetic marks, like methylations and histone marks, are removed and then restored back to baseline levels before offsprings are born. So how do genes escape these two barriers? So during the first reprogramming, which happens post-fertilization, there's a mechanism called methylation maintenance, where significantly methylated marks are actually protected by various molecules like the TRIM28 complex. An exciting finding was the HERA gene, which encodes a histone chaperone protein that aids in reprogramming. It was found to be a potential facilitator of TEI as its expression is correlated with something called transcriptional memory. This means that even if the parental methylation marks do get erased in later generations, the transcriptional changes associated with this methylation will still persist in the offspring. Lastly, contrary to previous belief, methylations can actually get erased and then remethylated in the same way later, allowing them to persist through this barrier without actually invading the initial demethylation, opening up so many more new approaches to studying this. Lastly, reprogramming happens yet again when primordial germ cells develop in the early embryo. Aside from identifying key facilitators of this process, we found that multiple studies examined the H3 lysine 4 trimethylation mark. It happens to be enriched in regions that are highly methylated and escape reprogramming. This opens up a very important idea that rather than every human having the same couple of genes that tend to escape reprogramming routinely, the parental genome and histone modifications can actually determine which epigenomic regions get inherited or not. Lastly, we evaluated these studies for the models they used and determined animals like chicken and mice to be less relevant models for human TEI due to their methylation mechanism differences, while something like the pig is one of the better emerging models to use. Yet, there remain many limitations in the current literature. It is very hard to know whether it was TEI or simply mutations or a shared environmental condition causing certain observed phenotypes in these studies, and there's still a lot of disagreement, with some studies still finding no evidence of TEI where these marks do get erased within 2 generations. To address these, future studies should optimize genome-wide long-read methylation tracking methods for more generalizable results, and use multi-omic technologies that basically look at the interplay of some of the important regulatory proteins and genes identified in this review. This could also help shed light on how different types of epigenetic marks interact with each other, as very little studies on TEI currently do so, and these marks can potentially also be inherited together. I would like to acknowledge my mentor, Natalie, for all of her help, and the SRI staff for organizing this amazing program. Thank you for listening.
Only you can decide what breaks you. As a child, these words meant nothing, but little did I know they would soon become my biggest mantra, helping me embrace my struggles. Hello everyone, my name is Trinika Gudlevan-Kadasiva, and I'm proud to say that my personal experience with gastroesophageal reflux disease is what sparked my interest in gastroenterology and led me to explore the differences between irritable bowel syndrome, IBS, and inflammatory bowel disease, IBD, for my literature review. Despite similar symptoms, IBS and IBD have distinct causes, treatments, and implications. IBD includes ulcerative colitis, which affects the colon and the rectum, and Crohn's disease, which can affect any part of the digestive tract. IBS, on the other hand, is a functional disorder without inflammation and has four types: diarrhea predominant, constipation predominant, alternating, and unsubtyped. Because IBS and IBD can show similar symptoms, it's often hard to tell them apart. This confusion often raises the question: could IBS be an early or late stage of IBD? My review aims to answer this question by investigating various studies on IBS and IBD to better understand their similarities and differences. Let's start with the symptoms. IBS typically causes pain that improves with bowel movements and lacks severe symptoms like fever or bleeding. In contrast, IBD pain is persistent, linked to inflammation, and is often accompanied by severe symptoms affecting other organs. Additionally, colonoscopy results for IBS show normal mucosa, while IBD shows inflammations and ulcers. This highlights that IBD has a more severe physical cause compared to IBS. When it comes to risk factors, IBS is often linked to childhood stress, anxiety, and infections. On the other hand, IBD is associated with smoking, urban living, and is more common in developed countries. And this is important because it indicates that IBS often has more psychological roots, while IBD has more environmental origins. In terms of diagnosis, IBS is diagnosed based on symptoms using the Rome criteria, while IBD requires invasive endoscopies. However, new research on non-invasive biomarkers for IBD like fecal calciprotein BAFF is gaining attention. These markers show higher levels in IBD compared to IBS in healthy controls, though optimal cutoff levels are still being studied. Thus, while not yet in clinical use, these non-invasive markers are promising for easier future diagnoses. In terms of treatment, IBS is managed with lifestyle changes such as the low FODMAP diet, cognitive behavioral therapy, hypnotherapy, and mindfulness based therapies, which are more beneficial for IBS than IBD. In contrast, IBD treatment involves medication to reduce inflammation. Recent research shows that probiotics can lower inflammatory proteins, reducing disease severity and maintaining remission in IBD. However, again, optimal doses and formulations need more study. Probiotics are less effective for IBS due to their minimal inflammation, and this again is important because it highlights IBS as more holistic or psychological causes compared to IBD's physical origins. Finally, irritable inflammatory bowel syndrome IIBS describes a condition combining features of both IBS and IBD. People with IIBS experience chronic stomach pain like bloating, altered bowel movements, and things like that that are akin to IBS, but they also have the mild gut inflammation seen in IBD. So, despite these overlaps, it is important to understand a key takeaway that IBS and IBD are still different conditions. They just happen to coexist like many other conditions do in the medical field. So, to ensure effective treatment, it's important to follow guidelines like the AGA Clinical Practice Update to differentiate between IBS and lingering IBD symptoms. So, again, in summary, while IBS and IBD may share some symptoms, their underlying causes and treatments are fundamentally different. And understanding these differences is crucial if you want to be able to provide patients with accurate diagnosis and effective management, ensuring that each condition receives appropriate treatment. Before I conclude, I want to extend my sincere thanks to the Harvard Open Biolaboratory Student Research Institute for this invaluable opportunity because it has allowed me to deepen my knowledge and passion for scientific research and medicine in a way I did not think was possible. And last but not least, a special thanks to my mentor, Ms. Susan Dang from Harvard Medical School. Her guidance and feedback have been crucial in improving my review, and I'm extremely grateful for her support. Thank you all for taking the time to listen.
My name is Mr. My name is Mr. Patrick, and I worked on analyzing and predicting temperature extremes using machine learning models. And then my professor was Professor Hiwank. I'm actually a student at Pembroke College. Okay, so I want to begin my presentation. 166,000 people died in 1998 to 2017 due to heat waves in the United States of America, 25% reduction in greenhouse in Russia in the year 2010 just because of temperature extremes. In Kathmandu, Nepal, the heat caused increased wear and tear on roads and buildings, leading to higher maintenance costs. In Burkina Faso, heat waves caused dehydration and heat stroke in Burkina Faso. So this study investigates the efficiency of various machine learning models in analyzing and predicting temperature extremes. It took temperature anomaly data from 1850 to 2024 because of the comprehensiveness of the data we actually needed for the model and to be able to analyze the changes in climate, identify trends, and train valid models. We took the data from four different areas in four respective continents. So Burkina Faso, we took Nepal, we took Spain, and then we took California. Because all these areas have actually experienced significant climate variability, irrespective of their geographic locations. We use Google Collaboratory because of its free access to computational resources, integration with Google Drive, and pre-installed libraries. We reprocess the data, replaced missing values with a simple imputer function, and then optimize the data. We split the data into the training data and the testing data in a ratio of 90 to 10 due to the complex nature of the data. And then we train four models in Google Colab, linear regression model, decision tree regressor, random forest regressor, and then a prophet model. We trained the model with our training data, then we tested the model with our testing data. And then when we were done, the values that we actually got, the prophet model and the random forest regressor had values that were closer to the actual values. But for the linear regression model and then the where decision tree regressor, their values were far away from the correct values. And so after that, we use six evaluation metrics to try and then evaluate the data and then check its potency. We use the mean absolute error, the mean squared error, the relative mean squared error, the R squared, the adjusted R squared, and the mean absolute percentage error. And then for the mean absolute error, the mean squared error, and the relative mean squared error, the first model had values ranging from 0 to 0.5. And then for the adjusted R squared and the R squared, it had value ranging from 1 to 1.3. And then for the mean absolute percentage error, it had a percentage of 0.35%, proving efficient amongst all the other four models, which had values that were not in good faith. So after that, we concluded that the profit model is the best model for the prediction for time series prediction. And so why is studying and predicting temperature extremes even crucial? This is very crucial for developing strategies to mitigate the impact of temperature extremes and then develop changing climate and be able to adapt to changing climate conditions. And so this is where my presentation ends. And I'd like to thank so much my mentor, Professor Hiwa, even though I was not efficient in machine language, he has been able to harness this skill out of me. And then looking forward, I plan on making very good use of machine learning in the future to come. I'm really grateful. And I'm also grateful to God. I'm also grateful to the SRI staff for this great opportunity given unto me to partake in this great initiative. Thank you very much. Bye.
Hi, my name is Prisha Bott, and I'm from Plano East Senior High in Plano, Texas. And this summer, I conducted a systematic literature review on the role of the gut microbiome in arsenic detoxification in humans. So, arsenic is a naturally occurring metalloid found in soil and water, and it possesses significant health risks, including cancer and cardiovascular diseases. In fact, the World Health Organization reports that arsenic contamination threatens the livelihoods of over 150 million people worldwide. And this issue is just becoming more prevalent because rice, a staple food crop, is more prone to arsenic contamination due to the increasing incidence of drought, which leads to the reuse of arsenic contaminated groundwater. And this topic is of interest to me specifically because I've been conducting wet lab research on arsenic contamination and its remediation from soil and crops. And I learned that even when different individuals are exposed to the same amount of arsenic, only some are more susceptible to arsenic related health complications. while others remain unaffected. And that's when I learned about the extensive research on the role of the gut microbiome in arsenic detoxification. Therefore, understanding how the human body detoxifies and metabolizes arsenic is crucial for developing effective interventions. Therefore, I conducted a systematic literature review focusing on the interactions between the gut microbiome and arsenic to contribute to insights for developing therapies to increase arsenic detoxification and resistance in humans. So using the Scopus database, I identified several articles of interest. And out of the 117 articles I initially identified, I ultimately chose 54 articles for my review. And here are some of my main takeaways. So the inorganic arsenic that's absorbed by the body undergoes a series of reduction and methylation reactions catalyzed by the enzyme arsenic reductase and arsenic methyltransferase, which converts the inorganic arsenic into various methylated forms, which are generally considered less toxic than inorganic forms, including monomethylarsenic acid, MMA, and dimethylarsenic acid, or DMA. So because these forms are more water-soluble, they're more readily excreted from the body through the urine. And arsenic-3 is the most toxic form of arsenic, meanwhile dimethylarsenic acid is the least toxic. And out of all of the gut microbiomes, bacteria and archaea are the most important in arsenic metabolism. And the most prevalent bacteria that we should remember are bacteroids, Clostridia, Lactobacillus, E. coli, Schwannella, and Bifidobacteria. So these bacteria possess a gene called ARSC, which encodes for the arsenic reductase enzyme. Meanwhile, archaea act as a methylating agent that converts arsenic into its less toxic forms. So probiotics are known to be able to supplement beneficial bacteria to the human gut microbiome. However, these probiotics cannot supplement archaea. So instead, what can be done is prebiotics can instead be used to proliferate archaea to improve the efficiency of arsenic detoxification. So in the future, the targeted supplementation of probiotics and prebiotics that promote arsenic metabolizing archaea could be a very promising therapeutic strategy. And another potential avenue of study in the future is the variation in gut microbiome composition across individuals from different geographic regions with different diets. And overall, this study highlighted the critical role of gut bacteria in reducing arsenic toxicity, thus guiding the development of gut bioremediation therapies. Finally, I would like to thank my mentor, Shannon Dixon from Ohio State University, and the SRI for giving me the opportunity to conduct my research.
Hello everyone, my name is Kayla Pang and I am a rising junior at the Kempley School in New Jersey, and today I'm really excited to be talking about my project, the socioeconomic impacts of sea level rise in the New Jersey Meadowlands. Global warming is obviously one of the biggest threats to humanity in the 21st century, and that is largely due to increased concentrations of greenhouse gases in the atmosphere. For example, in 2019, atmospheric concentrations of carbon dioxide were higher than at any other time in 2 million years. And it's because of trends like this that a warming of 1.1 degrees Celsius above pre-industrial levels has already occurred. This warming has severe consequences across the board, including sea level rise. For the Meadowlands area, a wetland within 3 miles of New York City, it is estimated that sea level rise will reach around 3 feet by 2100. In our literature review, looking at papers focusing on the ecological impacts of sea level rise in the Meadowlands area, we noticed that the invasive Phragmites australis, which is the main plant of the Meadowlands, is more vulnerable to sea level rise compared to its competitor, Spartina alterniflora. This implies an increase in biodiversity for the Meadowlands, since Phragmites is an invasive plant while Spartina is a native plant. And then we looked at literature attempting to calculate the socioeconomic impacts of sea level rise. And we distinguished two main categories: studies that focused on impacts to real estate and studies that focused on impacts to specific economic sectors, such as tourism. There is a fairly limited amount of literature calculating the socioeconomic impacts of sea level rise, so we decided to do a research project on this topic. Based on what I read, I had a few research questions. Number one, what is the value of real estate in the Meadowlands area impacted by sea level rise? Number two, what are the sectors in the Meadowlands area most vulnerable to sea level rise? And number 3, is there a premium for properties that are closer to waterfronts? To answer these 3 questions, I utilized LiDAR elevation data and reclassified their elevation increments to create 3 inundation maps showcasing which areas of the Meadowlands are going to be inundated at 1 feet, 2 feet, and 3 feet of sea level rise. And then I overlaid those 3 maps with parcel and land use datasets. Lastly, I used a nearest neighbor analysis to determine whether a property's proximity to water bodies was correlated with any real estate premiums. From this, I had 3 main key findings. My first key finding is that by 2050, $1.7 billion of property will be inundated, and by 2100, that number grows to $4.3 billion. You can see just how severe this economic consequence is for the Meadowlands area in the chart below, looking at how much of the Meadowlands is going to be inundated by 2100. My second key finding is that industrial and residential areas are more sensitive to sea level rise compared to ecological areas. And that is also apparent in the 2 pie charts below, looking at the green section representing commercial and industrial areas expanding from 1 feet of sea level rise to 3 feet of sea level rise on the right. My third key finding is that a property's distance to its nearest water body is negatively correlated to its sale price, which suggests that there is a premium for a property's proximity to waterfronts. These 3 key findings really point to the severity of sea level rise impacts on not only the Meadowlands economy, but the New Jersey coastal economy in general. Because of this, coastal authorities need to prioritize planning for rising sea levels by building seawalls, resilient infrastructure, etc. My study, by focusing on the socioeconomic impacts of sea level rise, the results appeal to many stakeholders and can lead to effective resource allocation. I thank the Harvard Open Bio SRI staff for this opportunity to present, as well as Dr. Peckman from the Meadowlands Research and Restoration Institute for her guidance throughout this project. Thank you so much for listening.
Hello, my name is Krishna Arun, and today I'm going to be speaking about my research project for this year. But before I go directly into the technical details of the research, I'm going to give you a little bit of a background of the problem that I'm solving, which is improving treatment planning for a specific type of brain cancer called glioblastoma multiforme. I chose this brain cancer to study as it is one of the deadliest diseases in the world with a 5-year survival rate of 5.6%. One of the biggest reasons that this disease is so deadly is because it grows very rapidly at a rate of 1.4% every day, being very invasive to important parts of the brain. And because the brain is so crucial to the body's function, so many people die so quickly given this disease. Common treatments are radiotherapy and surgically removing the tumor, also called resecting the tumor. However, even given the high stakes of this disease, doctors are not using advanced technologies to aid them for diagnosis and treatment. This is primarily due to minimal training data for technologies such as AI models to train from, which results in software companies not making software for this disease, basically forcing doctors to use old methods. So in my research project, I'm investigating if using AI will have a positive impact on modeling operation conditions accurately given an preoperative image with even small amounts of data. The first challenge when making any AI model, especially this one, is finding good data. In this case, it would be brain data of different tumor sizes and shapes and locations. So when given any preoperative image, it should be able to generate an accurate image of the intraoperative conditions. The REMIND dataset, which our software uses, does just that. Here are some examples in the first square of the methodology. One particular dataset that we have has many different MRI scans for the same patient. Each scan is like a filter on the same images used for highlighting different parts of the brain. In our case, the best type of MRI scans would be the gadolinium enhanced T1 weighted MRI scan, which highlights brain abnormalities like tumors. Our second decision was picking if the MRI scan would be in 3D or 2D format. The 3D format is made up of multiple 2D images around the brain, while the 2D format is in just one 2D image from the top of the brain. As we had budgeted only 4 weeks for programming the software, we had opted for the 2D MRI scan due to time constraints. Now let me talk about the AI model we used, which you guys can follow in part 2 of the methodology. Our software is built on a modified version of Monai's generative model, called diffusion model UNet. This model is specifically designed for 2D MRI slices, and it leverages Gaussian noise as a key component of its image generation process. The model takes in 2 input channels and outputs a single channel to predict post-operative outcomes. It consists of 3 stages, each with 64 feature channels, using ResNet blocks for downsampling and upsampling. Gaussian noise is added to the input image, transferring them into a noisier version. Then the model learns to reverse this process. In the reverse phase, it incrementally removes the noise, using the UNet's skip connections and convolutional layers to reconstruct MRI images. We had trained our model for 800 epochs and validated our software using the training and validation graph and the SSIM score. The training and validation graph, which you can see in figure 1, mainly allows us to see if our model is overfitting or not, which is crucial given that we only have 40 data points. When evaluated, we had found that our model, even with the low amount of data performed, with the same overall curve as the training and validation graph, knowing that it does not overfit and also generalizes pretty well. The SSIM score can be seen in figure 2, which compares the similarity between the generated and validation images. On average, our model got a score of 0.30, with individual scores ranging from 0.18 to 0.44. Although our score is relatively low, given that our data set had only 40 data points, it's a big deal that it did not score in the negatives. Because of this, we could conclude that our model could successfully provide doctors with a simulation of operative conditions, even when having small amounts of data and not overfitting, giving doctors a positive impact in modeling operative conditions. Future research would focus on getting higher resolution images and factoring brain geometry. With these changes, the model can more easily attain the uber goal of being able to model full resectional and partial resection images more accurately with segmentations as an added input, so can be able to model tumor growth. A flowchart can be shown in the right side of the poster. With these changes, I know that our software can change the world. Thank you.